Good day, this is Professor Will, CBMD PhD, and today's Tuesday, June 7th, 2022. It is 4:27 and 46 seconds, AM Eastern Standard Time, and today I'm going to be doing a research lecture on cancer, so let's hop to it here. A promising compound kills range of hard-to-treat cancers by targeting a previously undiscovered vulnerability. Date June 2nd, 2022. Source: University of Texas Health Science Center at San Antonio. A compound called DRX41 targets a previously unrecognized vulnerability and difficult to treat types of cancer, including triple-negative breast cancer. The compound will be studied as a drug for clinical translation. Researchers identified a critical vulnerability in multiple cancers and have validated their findings in multiple cancer cell types and animal models. The range of cell lines and xenografts in which the compound has been shown to work is compelling and indicates that it is targeting a fundamental vulnerability in cancer cells. Xenografts are human tumors grown in mouse models for research purposes. The laboratory studies breast and ovarian cancer progression, including therapy resistance, with a goal to developing small molecule inhibitors for therapy-resistant cancers. In 2017, researchers and colleagues identified a compound called DRX11 that targets the estrogen receptor, a protein that drives the vast majority of breast cancers. From a screen of chemical analogs of ERX11, the researchers identified a compound called ERX41 that only linked ER-positive cancers in petri dishes, but also readily killed triple-negative breast cancers, including more than 20 distinct triple-negative breast cancer cell lines. Triple-negative breast cancer is a cancer subtype lacking receptors for estrogen, progesterone, and human epidermal growth factor 2 and 4, which there is a paucity of targeted treatments. The researchers expanded these studies to show ERX41 had activity against a large number of human tumors grown from several of these cell lines in mouse models. In addition, ERX41 was potent against patient-derived xenografts as well, causing shrinkage of these human tumors grown in mouse models without affecting normal breast cells or causing any discernible toxicity in these animals. The safety profile and high therapeutic index of this compound is particularly notable and bodes well for clinical translation. Other experiments show that, in addition to ER-positive breast cancers and triple-negative breast cancer, ERX41 is also effective against other cancer types with elevated endoplasmic reticulum stress, including pancreatic, glioblastoma, and ovarian cancers, which all have few effective treatments. The endoplasmic reticulum is a structure in many cell types that performs assorted functions, including manufacture of proteins. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.